Vish, you can stay if you want. If not, I'm going to go ahead and start this review with them on chapter 2 and 3. So we're doing chapter 2 and 3 today. So this basically is talking about enzymes. So, and we're just going to go through some of the things. Sucrase, for instance. What you want to know for sure is when you're doing your enzymes, look at the prefix. Look how it starts. So like sucrase breaks down sucrose. If you see a protease, then you take that first part of it and it looks like protein, right? So it looks like protein. So when you see that, a protease is one of those things that can break down protein. And of course, lipase, and so lipase will break down lipids. Okay, everybody follow me there? Now, here's the other thing, is that, and I know that this is crazy, if it sounds like a duck, it must be a chicken. Well, that's one of those situations where a lot of times the enzymes themselves will tell you what they do. For instance, let's see, and when you get ready to answer a question, turn your microphone on and then answer the question. So I'll ask John, what is the function of a kinase?
To add or remove a phosphate group.
Okay. All right. You said to add or remove a phosphate group. Let's see if he's right. Remove or add a phosphate in the presence of what? ATP, ADP, or AMP. Okay. So if we see that, then we know that the enzyme that's responsible is going to be a kinase. What is a dehydrogenase, Ambria?
Sure.
Okay. How about, let's see, who else is on here? Moment, what is the function of a dehydrogenase?
Is it to take out hydrogen?
Okay, we can kind of live with that. So a dehydrogenase is also what we call an oxidoreductase. So it falls under the category of an oxidoreductase, but it's involved in redox reaction. So basically, like you say, removing those hydrogens, which you'll see anytime that you see, for instance, NADH, FADH2, those are always going to be associated with a dehydrogenase. Everybody understand that? Good. Perfect. Now, at any point, if you don't understand something, you can raise your hand and then just turn your mic on, okay? Now, of course, the next thing was an isomerase, and we should all know what an isomer is. Now, in cellular respiration, what isomers do we talk about that we mentioned on yesterday? Raise your hand if you know. Okay, I got two people. John, you came first. So, John, go ahead.
Glucose and fructose.
Okay, very good. Glucose and fructose. Very good. So glucose and fructose are those two things. Those are isomers, both C6H12O6. Good. What does a phosphatase do? A phosphatase. Okay. So a phosphatase, what it's going to do is going to remove inorganic. Okay. Oh, Donish was typing. So it's going to remove inorganic phosphate, basically. So the removal of phosphate in the presence of ATP, we said would be a kinase. But if it's an inorganic phosphate, it's going to be a phosphatase. If we're adding an inorganic phosphate, then we're looking at something like a phosphorylase. Okay? I got that? All right. So let's look at this next one here, which talks about. Read this question for me, Moment.
If carbonic anhydrase were put in a solution with a constant decreasing pH, what would happen to the CO2 concentration in the lungs?
Okay, gotcha. So you see the answer right there, Moment, right?
Yes.
Yeah. So CO2 in the lungs would decrease. Now, here's my question. Since the CO2 in the lungs would decrease, can someone explain Why? Why would that decrease? And you can just raise your hand if you think you know why it would decrease. Okay. Go ahead, John.
CO2 is basic, so in a decreasing pH that's becoming more acidic, the CO2 will decrease.
Okay. So, okay. But why would it decrease in the lungs, though? Now, let's look at something. So you see I have three principles here, right? One is concept, application, and critical thinking. The concept is what? The concept is how are enzymes altered? Because if you notice, carbonic anhydrase, you see the ASC at the end, indicates that it's an enzyme, right? Well, it's an enzyme that's put in some type of solution that has a consistent decreasing pH, so that pH is lowering. So what they're trying to do sometimes, they may even do this on the exam, is actually test you on a basic science idea or thought, such as an enzyme in an acidic solution or an acidic environment. What happens? Well, some enzymes love an acidic environment. Some don't. But you want to pick up here is that pH can alter the effects of an enzyme. Everybody got that? Hello?
Yes.
Okay. All right. Good. Okay. So even if you, like I said, if you want to say, you can say yes, you can type yes, or you can simply just click the agree sign, and then that'll let us know, that you agree. That way, I kind of know who is following me. So if you click the agree sign, that means that you understand what I'm talking about. If you don't, just raise your hand. Now, if this is what we're looking at, if we're looking at how are enzymes altered or what happens to them, the application to that question would be altering this enzyme has what effect on CO2? Okay? So here's the critical thinking piece. First of all, in order to understand this, you first need to know what is carbonic anhydrase. How does carbonic anhydrase work? And does anyone know? Okay. So no hands going up. Nobody's raising their hands at all. So that means I'm going to have to pick on somebody. Ambria.
Assuming it removes something.
Okay, remove something. Okay, all right. Okay. Donish. Remove CO2. Okay. John or Momin, you have any other thoughts?
It removes.
Go ahead. Go ahead. Go ahead. Go ahead, John. Go first.
Does it convert CO2 to bicarbonate in the blood?
Okay. All right. So John says it converts CO2 to bicarbonate, and so what that means is that CO2 is transported through the blood in the form of bicarbonate, HCO3, right? HCO3 minus. Well, here's the deal. So if you know that's what it does, then your next question, your next critical thinking piece would be, what is the relationship between CO2 and the lung? So, Moment, can you tell me the relationship between CO2 and the lung?
CO2 is released from the lungs.
So it's released from the lungs. Where does it go?
Out of the body.
Okay, I see what you're saying. Where does it come from?
Comes from cellular respiration.
Okay, all right, okay. So, okay, you're on the right track. So basically what CO2 basically, and he is right, you do get during exercise and things like that, you get a buildup of CO2, which can go towards lactic acid. So when you're exercising, this starts to build up in the tissues, right? But we know that the tissues are cells. That same tissue, that same CO2 that builds up, once it starts to collect in tissues, can cause cramping and things like that. So what you want to do is you want to transport that CO2 away from the muscles and towards the lungs so that you can blow it off and exchange it for CO2. And we'll actually talk about that in respiration. But basically, it's transported through the vascular system. And once it gets to the pulmonary artery and then enters, so the pulmonary artery carries what type of blood, Ambria? Hello? Did you say something, Ambria?
It doesn't carry oxygen in the blood?
Okay, so you're saying that the pulmonary artery carries deoxygenated blood, right?
Yes.
So then that deoxygenated blood, but it's carried in the form of bicarbonate, right? So some kind of way we got to convert that bicarbonate to CO2 because you have to blow the CO2 off. So what do you think we need in order to do that, Donish? And just for speed, if you don't know, just click or just say no, or you can chat no. That'll be quick. IDK. Okay, good. So what do we need for that, John? What do we need in order to do that?
Bitters?
Yes, good. So we need carbonic anhydrase. So with that said, if carbonic anhydrase is not functioning like it should function, that means that when it's time to dump our CO2 and we're supposed to transport it into the alveoli, if we don't have that, then what happens to the concentration of CO2 in the blood? Uh, let's see. Who says increase? If you think it increases, the amount of CO2 in the blood would increase. Check. Okay. That's two. Two people checked. Donish? And Bria? Now, either you're going to have an answer, you're going to either check. So, Donish, use your check for me. That way I know if you know. So Donish disagrees. Wait a minute, wait. So, Donish, are you saying that the concentration of CO2 would decrease in the blood? Or the concentration of CO2 would increase in the blood? Increase. Okay. So everybody agrees, basically. Okay. Okay, Donish has a question. Go ahead, Donish. Oh, okay. All right. All right, so everybody understand that? Let's go to the next one. The next thing we're going to be talking about is cooperative binding, okay? When you think about cooperative binding, think about cooperating. That's when one of us binds and we all want to bind even tighter. An example of cooperative binding is going to be that which you see with hemoglobin. So, hemoglobin has these four subunits: alpha and beta, alpha 1, alpha 2, beta 1, beta 2. And so, say for instance, oxygen binds to the alpha 1. Then what that's going to do is increase the affinity of for oxygen to the alpha 2 and beta 1, beta 2. Does everybody understand that? Good, good. Okay, good, perfect. Okay, so everybody got that. Okay, so what will a cooperative binding curve look like? Let's see. Moment. What do you think the curve would look like? Would it be linear? Would it be sigmoidal? Would it be, I mean, what kind of curve?
I assume it would be exponential.
So you think it would be exponential. So you mean, so if I drew it, let me ask you this. So basically, would you say that the curve, um, so what you said, would the curve look like this moment?
No, it would like excuse me.
You think it would look like that?
Yep, the other way.
Which one? Let me mark them for you so you can see the difference. So we're gonna say this one, you got this one, this is B, and this is C. These are bad curves, trust me, they're not beautiful. But which one is it not for sure?
B.
Okay, it's not B. So you think the curve would be more what, like sigmoidal?
What does sigmoidal mean?
Okay, good. Okay, I'm glad you asked that because John was ready to answer. Go ahead, John.
S-shaped.
S-shaped. So John says it's S-shaped. So John, why would you have a sigmoidal curve in cooperative binding? And why would it not be like a linear?
Because the affinity for hemoglobin isn't constant.
Okay,
that's why it
good.
wouldn't be linear.
Okay, all right, okay. Everybody understand that? So it would be sigmoidal. So, all right, so what you see here, you see this enzyme? This is what we call Michaelis-Menten. And Michaelis-Menten looks at enzymes and how enzymes function, basically. Let me stop the drawing off. And so what you see here is competitive inhibition and non-competitive inhibition. Competitive inhibition means that you have an enzyme with an active site. A substrate can bind to the active site. But in competitive inhibition, you can have a competitor that can also bind to the active site. Okay? Now, the second part to that is, in non-competitive inhibition, you have a substrate, and it still wants to bind to the active site. But now you also have a non-competitor that will bind to what we call an allosteric site. That's other than the active site. And when it does that, it changes the conformation of the active site. And when you do that, that original substrate has either true difficulty binding or has a limited binding. Now, we're not going to talk about the limited binding because you can still actually have some binding of substrate, but in it's modified. They're not going to probably ask you about that on the MCAT. So, the question is, if you have a non-competitive inhibition, and I want you to understand this, as long as the active site, this is one easy way to remember it, as long as the active site is occupied, whether it's with substrate or whether it's with a competitor, you will eventually and can eventually reach Vmax. Okay? However, if you have a non-competitive inhibitor, then what's going to happen to your Vmax is going to decrease because you have limited binding of substrate. That's just a way to remember. Does everybody get that? All right, I'm going to keep moving. We'll be asking you questions soon. All right, so in cellular respiration, so glycolysis happens in the cytosol, right? The mitochondria pretty much does everything else. I mean, if you think about beta oxidation, Krebs cycle, electron transport, for the most part, your default answer should be the mitochondria, if you don't know. If it's not glycolysis, it's involved in cellular respiration, and you have no clue, then definitely pick mitochondria as your default. Okay? Now, if red blood cells lack mitochondria, How many ATPs are produced in aerobic respiration? How about Donish? Okay, no answer, Donish. How about Ambria? Now Donish is typing. Donish says 2. Ambria says 2. John? 2. Moment?
2.
2. Now, everybody agrees with that, right? And I guess Vish, you agreeing with that? Oh, good, good. So everybody agreed with that, except your instructor. Your instructor didn't agree with that. Now, before I tell you the answer, would somebody like to rethink it? Or maybe I'm wrong, maybe Vish is wrong. So would somebody like to rethink it? And once you know or have a thought, raise your hand. Okay, so nobody wants to rethink it. Okay, all right. Well, keyword, there you go. Keyword is aerobic. He gave y'all a hint. He did more than I was going to say. Vish, you want to go ahead and tell them the answer? I see typing. Okay, so all right, so I see where Vish went with this too. Okay, so let's follow this. Now, he would be right, but I guess I got everybody tricked today. I tricked everybody. So read the question very closely, everybody. If a red blood cell lacked mitochondria, so if you don't have mitochondria, then you are not able to undergo what? What processes could you not do without a mitochondria? Raise your hand if you know. Moment. Okay. Go ahead, Moment.
Cellular respiration.
Yeah. Well, no. You can still do cellular respiration, but what part of cellular respiration would not happen? John? I don't know. Okay, Ambria? Ambria's typing. She says oxidative phosphorylation. Okay, I like that. So Ambria said you could not do oxidative phosphorylation. Well, that's true because oxidative phosphorylation occurs where? In the electron transport chain, right? Well, we know that substrate level phosphorylation occurs where? In glycolysis. So let's look at the key parts of this question. First, we don't have mitochondria. Second, how many ATPs are produced? Not the net ATP, but how many ATPs are produced? So does anybody have a answer that they think is right? Moment. Okay, Moment, go ahead.
38.
38. Anybody agree with that? Disagree? John says 0. All right. So actually, the correct answer to this question is that you actually have 4 ATPs that are produced, right? Produced. Now, the net ATPs that you would have as a result of this would be how many? Well, four is what you produce. And so actually, how many ATPs do you use exactly? How many ATPs do you use during glycolysis? You use how many? Two, right? So if you produce four, you use two of them, it gives you a net of two. Donnie said 8. Okay. Donnie, I don't know where 8 came from. You're going to really have to help me with that. Okay. Does everybody understand what we just went through? Do you understand how that question was written and what was said? All right, so I'm going to keep going since nobody's raised their hand. Okay, now let's look at this question. If we identified that in the cytoplasm of a cell there's 20% adenine, then how much guanine would be present, and would this be a prokaryotic or a eukaryotic cell? Raise your hand if you think you know the answer. If you don't raise your hand, I'm going to probably pick on you. I'll pick somebody else. Okay? John, go ahead. John? Or you want to type it? Okay. So John says we have 30% G. All right? Show me if you agree with John. You just click your agree button. Is everybody getting comfortable with their buttons yet? Okay, Moment agrees. Ambria. Donish does not. Okay, Donish, you can disagree. There's a disagree button as well. Okay, how did he get 30? Okay, John, go ahead and tell us how you got 30.
How that adenine pairs with thymine. So you know that we're going to have 20% of that. That's 40%, and so that leaves us with 60%. And so we split, it's 60% guanine and cytosine, so half of 60 is 30.
Oh, gotcha. Okay. So, Donis, did you hear what he said? Okay, perfect. Okay. So, what's the second part to this question? Okay, Moment had a question. Go ahead, Moment.
I just, for some reason, I can't see everyone's chat.
You can't see everybody's what?
Chat.
Okay, can you? I'm not sure everybody can. You can see. Can you see my chat, Moment?
No, sir.
So you can't see any chats?
No.
Okay. Huh. I'm not sure what that is. I'm not sure on what that is on there. So, but we're not actually chatting that much. So can you hear everybody though when we speak? Okay, you can. Good. Okay. All right. And then we'll try to fix that later. All right. So, can somebody tell me whether or not this would be a prokaryotic or eukaryotic cell and why? No hands raised? Take a shot. It's a 50-50. Thank you. Now, John is not. I'm not going to let John talk. John is not going to answer all the questions. Go ahead, John.
My guess would be prokaryotic because its adenine was found in the cytoplasm.
Ah, all right. So, does everybody agree with that? Okay, Moment says yes. Donis, yes. And Bria, yes. Okay. All right. So, we know that there's a difference between a eukaryotic and a prokaryotic cell, right? Well, the answer is 30%. Great job on that. It is a prokaryotic cell, and we do know that in a prokaryotic cell that they don't have any membrane-bound organelles, so they don't have a nucleus. Since they don't have a nucleus, that means everything typically happens in the cytosol. Okay. Now, here's some terms we just need to know, and I'll just go through these. Nucleotide is a sugar base and a phosphate. What is the minimum codon size to satisfy 20 different amino acids? It's going to be 3. And we know that 4 to the third, because you have 4 different codons, 4 different nucleotides. 4 to the third is going to give you 64, but if you did like 4 to the second, that would only give you, like 16. So we know that you have to have at least a codon size with 3 to get those different amino acids. The nucleoside is just a sugar and a base. A pyrimidine, those are adenine and guanine. I'm sorry, that is wrong. That is totally wrong. What are my pyrimidines? Raise your hand if you know. Everybody, okay, thank you. Okay, Moment, go ahead.
Cytosine and thymine.
Good. What if I had RNA?
Cytosine and uracil.
Okay, good. Everybody understand that? Everybody's okay with that? So disregard this. So then obviously, pyrimidines is cut the pie, purines are pure as gold. That's the mnemonic. So disregard that. Ribose. Ribose is a 5-carbon sugar. And ribose is a 5-carbon sugar that you actually find that's developed or produced in what we call the pentose phosphate pathway. Pentose meaning 5. And that pathway actually comes from glycolysis, from glucose 6-phosphate. So you can actually go from glucose 6-phosphate to this ribose sugar to produce those different portions of the nucleotide. Okay? And then finally a histone. The histone is just a protein, right? Made of basic amino acids. And what it has is a positive charge around it. So since a histone is made of these basic amino acids, how is it that DNA is able to stay wrapped around these histones, Ambria? Okay. Let's see. Donish, can you type me and tell me how the DNA is able to stay wrapped around those histones? Not sure. Okay. So does anybody know? Raise your hand if you do. John or Moment? Okay, good. Okay, go ahead, John.
The histones are basic and the DNA is acidic, and they bond through hydrogen bonding.
Okay, so the histones basically have a positive charge, right? An overall positive charge, but the DNA itself has a what kind of charge? Does DNA have a negative or a positive charge? Okay, so somebody says negative. Okay. Okay. Negative. Okay. So everybody kind of see how that works out. Okay. Here we're talking about genetic mutations next, okay? So in genetic mutations, you have what we call, there are basically 2 genetic mutations. You have a point mutation, and then you have a frame shift. Now there are different types of frame shifts, and there are different types of point mutations. But here's what you need to know. First of all is the missense mutation. A missense mutation means that there's a change in one of the nucleotides, a single change. And that single change in that nucleotide will cause a different amino acid to be produced. That's called a missense mutation. A nonsense mutation is basically a mutation where you have, once again, it's a point mutation, but you have a single nucleotide chain. So here you have UGG, and you go from UGG to UGA. Now here, when you produce UGA is one of our stop codons. So anytime that you produce a stop codon, that's called a nonsense mutation. So one way to remember a nonsense mutation is non-stop. So nonsense mutations basically are for stop. So anytime you see a stop codon, that's introduced, okay? So your stop codons are UGA, UAA, and UAG. You go away, you are away, you are gone. Those are your stop codons, okay? Your silent mutation makes a lot of sense. Means that you still have that single that single chain, which AAG is now AAA, but the amino acid stays the same. In that situation, that's called a silent mutation. A frame shift mutation is one where you have a long row of, say, nucleotides, and you either take one of those away or you add a new nucleotide to the sequence. When you do that, it's going to shift the entire frame, the entire reading frame. When you do that, that's called a frame shift mutation. You can have that due to an insertion or a deletion. Any questions on that? All right, because I will be asking you questions. All right. Ambria, read this for me.
2 patients enter your clinic today. One with sickle cell anemia caused by the replacement of amino acids, glutamic acid with valine. The second patient has cystic fibrosis caused by a frame shift mutation, which is more lethal and why?
Okay. You want to answer it? Okay. Raise your hand if you want to take a shot at it. Anybody want to take a shot? All right. Go ahead, John.
Cystic fibrosis is more lethal because it's a frame shift mutation. So it alters the reading frame.
Okay. Okay, it does alter the reading frame. And so just because it alters the reading frame, does that make it more lethal? What is it in particular that makes this more lethal? And I will agree with them that cystic fibrosis is more lethal. But why? What is it about that's happening? So you remember in the first slide I was telling you all about three things to remember. And let me just show you what I'm talking about. So you remember I told you in the first slide that what you want to do is you want to remember 3 things. You want to remember concept, application, and then the final thing was critical thinking. Okay, does everybody remember that? What I was talking about? Those 3 things? So, good. So what is the concept here? What is the big concept that you should understand? Because the MCAT is testing you on whether or not you understand concepts. So what's the concept here? Raise your hand if you think you know what the concept is. Is the concept making turkey burgers? Uh, Anybody want to respond? Okay, I need some activity. I need something going on. We need to have movement. Is there life on this computer right now? Am I frozen? Okay, good. So you all are present. I just want to make sure. So the concept. What do you think is the big picture here? Do you think the big picture on the MCAT is for you to understand what cystic fibrosis is or what is sickle cell anemia? Do you think they're really testing you on your understanding of these diseases? Okay, good. All right, so I have 2 people to say no. Good. So no, they're not testing you on that. So if you know that that's not what they're testing you on, then what you can cook, what you can do, pick from this information, and you can actually develop your concept based on, since you know they're not testing you on sickle cell. Okay, good, perfect, John. So John said that the concept here is genetic mutations. Okay, good. That's the big thing. Do you understand genetic mutations? Do you understand kind of like what they are, how they function, that kind of thing, right? And so since that's the first concept. The application part is that now, okay, I know what genetic mutations are. I know you got point mutations. I know you got frame shift mutations. Now, how do I apply that information that I know about genetic mutation to this question where they're talking about two disease processes? Well, the application is that I have to identify whether sickle cell anemia is a frame shift or a point mutation. And we know that it's a point mutation, right? And what type of point mutation is this moment? What type of sickle cell anemia? What type of point mutation is that?
Yeah, frame shift mutation.
Did you say a frame shift for sickle cell anemia?
Oh, sorry, I meant the point mutation.
What type of point mutation?
The missense.
Perfect. Great job. Absolutely great job. So, so everybody understand what Moment said. He says that this is a missense mutation because he said that the amino acid changed. So since you know that, but it's still a point mutation. Well, you know, point mutation is sickle cell anemia. Cystic fibrosis, they've already identified, is a frame shift mutation. Anytime that you have a frame shift mutation, then what happens is that the protein itself is actually changed, or sometimes it doesn't even function. So sometimes, a lot of times, you get a non-functioning protein. So that's one of the things you see with a frame shift mutation. There are many conditions where you can see frame shift mutations. And a lot of times, frame shift mutations will show up very early in life. So it's babies and things like that. So when you are on the pediatric ward, like you start doing pediatrics, you're going to see kids who come in, and you're going to have kids who are having, say, cystic fibrosis, which basically, cystic fibrosis is just a defect in this gene we call a CFTR gene. And that gene basically causes a defect in the chloride channel. Now, that all of that can start messing up anything with a tube. Anything in your body that has a tube will be messed up. So the small intestine. So if you have a defect in the small intestine or the lining of the small intestine, what are some of the things that you might see? Embryo. Either click I don't know or say something. Okay. Darnish, do you know? The question is that with cystic fibrosis, it's a defect in the chloride channels, but it can involve the anything with a tube. So typically linings, things like that, like the small intestine, is one of those areas that's affected. So what are some of the things that you might expect to see uh, as a result of this in a child? Just name something you would expect a child to present with or to have. Loss of appetite. Okay. Okay. I can live with that. I wish you could talk because I'd love to hear why you say loss of appetite, but I can follow that. Um, okay. Well, yeah. So, okay. I'll let Donis finish typing and I'll read it. You said it conflicts with the smallest. That's right. Yes, that's true. So Donna said basically it alters or it affects the small intestine or anything with a tube. Good. So as a result, let's see. All right. Now that you must be typing a paragraph. Okay. Isn't the small intestine where nutrients and food are reabsorbed? Yeah, good. Now, Darnish did a great job. So he said that the small intestine is where you reabsorb a lot of your nutrients and things like that, right? Perfect. And so when the question was if the small intestine is affected, the concept is do you understand digestive system? That's the concept. Application is how does damage to the small intestine affect whatever we talked about? Where if you damage the small intestine, you have decreased reabsorption of nutrients. Now that can even manifest in decreased energy, and I'm sure you know why decreased energy. Because if you're not able to reabsorb glucose, blah blah blah, y'all understand, right? Okay, everybody got it? Oh, vicious type. I'm going to have to step. Okay. All right. So Rich said goodbye. He'll talk to you soon. All right, Rich. All right. So the answer was cystic fibrosis. It alters the protein and makes the protein function less viable. Okay. All right. Now, what I want you to do is I'm going to share a link with you. Hopefully, you can you can pull it up. Let me see if I can do it. Let me see. Web link. All right, give me 1 second. I want to see if I can share this with you. And then once you actually see it, I want you to be able to tell me. Okay, so I'm going to share this link, and I want to see if you can see it. Now, tell me if you can see the. Let's see if it's letting you see. Okay, can you all see the link that I shared? Okay. Are you able to click on the link? Okay. Good. Once you click the link, I want you to go to the link, look at the video. Once you're done looking at the video, come back and just say, just let me know you're back. Just say back.
The polymerase chain reaction is a method for making many copies of a specific segment of DNA, starting with a very small amount. This technique can be used to identify specific microorganisms from a small amount of DNA, and to identify persons involved in crimes from DNA on cigarettes or in a single hair follicle. The DNA to be amplified is mixed with deoxyribonucleotides, a thermal stable DNA polymerase called Taq polymerase, and DNA primers. The DNA primers hybridize to the ends of the gene to be amplified and provide a starting point for the Taq polymerase. The mixture is heated to break the hydrogen bonds in the DNA, forming single-stranded molecules. The mixture is then cooled sufficiently to allow the DNA primers to anneal to each end of the segment to be copied. Taq polymerase then synthesizes the complementary strand of DNA using the primer as the starting point. The temperature is raised again to separate the DNA strands and then lowered sufficiently to allow the primers to attach. Taq polymerase now synthesizes another set of new complementary strands. This process is repeated until enough DNA has been produced to be identified or used for further research. After 21 cycles, one molecule of DNA can be amplified to over 1 million copies. This amount of amplification can be achieved by running the reaction overnight in a thermal cycler, an instrument that automatically raises and lowers the temperature at appropriate time intervals.
Okay, great. Okay, perfect. Okay, so as you saw, that was PCR, right? And so polymerase chain reaction. Now, in a polymerase chain reaction, what is the advantage of PCR?
The polymerase chain reaction is a method for making many copies of a specific segment of DNA, starting with a very small amount. This technique can be used to identify specific microorganisms from a small amount of DNA.
What is the advantage? What is the advantage of PCR? So what is the advantage of PCR? Okay. Go ahead, Moment.
It helps in crime scenes.
Okay, helps in what?
Crime scenes.
Okay, but good. Okay, so it does help in crime scene. Donish, you say it picks out specific organisms. Well, the true advantage of PCR is that you're able to take a small piece of DNA and replicate it. So you don't need this huge DNA library to figure out what the genetics behind this is. So that's one of the advantages of PCR. Okay. Now, what if you were doing PCR? What would be something that would inhibit or alter that could affect your PCR results? And just raise your hand if you know. Something that could affect or alter your PCR results. So you're in the lab, you want to do PCR. What is something that if it happened, it could alter your PCR results? Nobody raising their hand? Nobody want to take a shot? Okay, well, let's look at it. Do you all agree that PCR requires increasing temperatures? Okay. All right. I got Ambria, Donish to say yes. What about John and Moment?
Both increase and decrease.
Okay, good. But you definitely need an increase in temperature because increasing the temperature allows the strands to break, right? Then, if that's the case, what if we could not raise the temperatures high enough? Would we still be able to get an effective PCR? Okay. Okay. All right. So I got a no, and a no, and then Moment. No. Okay, gotcha. Okay. So the same thing with the cooling temperatures. So if the temperatures are altered, those things can alter your PCR results. Is everybody follow me? Sounds good? Yes. Okay. So the next thing that we'll be looking at is the concept of operons. And one of the things I was asking you all to do is look at the repressible and the inducible operons. So we're going to look at the first one we're going to be looking at is one that's called the lac operon. And so the lac operon is pretty cool. So we'll be taking a look at that one first. And then I'm going to put it in here so you can see how that one works. Now my question is, let's see, is the lac operon, is it a repressible or inducible operon, Ambria? She says it's repressible. Do you all agree with that? John says no. So John, can you tell us, cut your mic on and tell me what you think?
I think it's inducible.
Okay, good.
Because
So he says it's inducible. So if that's the case, then let's take a look at it. So I'm going to share this link with you, okay? And when I share this link, I want you to take a look at it. And then once you see it, go ahead and click on it. And just like we did before, just tell me, when you're done, just say I'm done, okay?
Many bacteria grow on glucose before they utilize other compounds, such as lactose, as a growth substrate when both are present in the medium. Transcription of the lactose operon is controlled by two regulatory proteins. The catabolite activator protein can bind to the activator binding site and facilitate transcription, and the lac repressor protein can bind to the operator site and block transcription. The catabolite activator protein can bind to the activator binding site only if cyclic AMP is bound to it. When glucose is present, the concentration of cyclic AMP in the cell is low. RNA polymerase cannot bind efficiently to the promoter, and transcription does not occur unless the catabolite binding protein is bound to the activator binding site. Therefore, transcription of the lac operon does not occur in the presence of glucose. In addition, if there is no lactose in the medium, the lac repressor binds to the operator site, and this blocks transcription. When both glucose and lactose are present, there is insufficient cyclic AMP to bind to the catabolite activator protein, which therefore cannot bind to the activator binding site. Without the bound activator protein, RNA polymerase cannot bind to the promoter and initiate transcription, even though lactose binds to the repressor and prevents it from binding to the operator site. When neither glucose nor lactose is present, the concentration of cyclic AMP is high, and the catabolite activator protein is bound to the activator binding site. RNA polymerase can bind to the promoter, but it is blocked by the repressor on the operator site. When glucose is absent and lactose is present in the medium, cyclic AMP is present and binds to the catabolite activator protein, which in turn binds to the activator binding site and facilitates transcription. Lactose binds to the repressor and prevents it from binding to the operator site.
Just let me know you're done.
RNA polymerase can now bind to the promoter and carry out transcription.
So Moment is done, John is done, Donis is done. Just waiting on Ambria. Okay. So, Moment, can you describe the process of the lac operon, how it works? Go ahead and turn your mic on.
If there is glucose, there's low cyclic AMP. So the uh, the diamond, the yellow diamond in the picture, it can't attach on.
Okay, you said the yellow diamond. Does it have a name?
I have to play the video.
Okay, all right, okay. Okay, you got to start it. John, kind of help out a little bit. Let's see what you got.
I think was the diamond the repressor?
Well, the repressor was like the horseshoe.
Okay. Well, the lac operon is turned on in the presence of lactose. Which will inhibit transcription, I think.
Okay. Well, let me ask you this, John. What do you see on your screen right now? You just see what? Can you see my screen? John.
Just the link, you mean?
Is that so? When I clicked over, all you could see was so all you saw was all you see is the link, right?
Right now, yes.
Okay, good. Okay, so you see the link. So can you hear me, John?
Yes.
Can you hear me now?
Yes.
Okay, perfect. So I'm just trying to see something. I'm testing something out. Can you still hear me, John?
Yes.
Okay, good. So, John, you remember in that lac operon that basically what happened was you had cyclic AMP, you had a protein, and then you had a repressor, right?
Mm-hmm.
And so what happened is that basically if you had glucose present, if glucose was present, then what was going to happen is that you had an RNA polymerase, right? If glucose is present, it cannot bind to the promoter region. With me?
Uh-huh.
If that's the case, then the repressor, right? That repressor basically comes in. So when you have glucose and say lactose present, you don't have enough cyclic AMP to bind to the activator protein, which will mean that you cannot bind to the activator binding site. So if that's the case, if you cannot do that, if you don't have an activator protein, then RNA cannot do what it needs to do at the promoter, and it cannot initiate transcription. So what is the take-home message? Is that this is a lac operon, right? If you have RNA polymerase, and it can bind to the promoter, but you block it with the repressor, right? So what would happen? Ultimately, what needs to happen for transcription to occur? Uh, John, you still there?
You'd have to get rid of the repressor.
Okay. So if glucose is there, is cyclic AMP high or low?
Low.
Gotcha. Cyclic AMP is a second messenger, right?
Yes.
So if that's the case, if cyclic AMP is low, do you think transcription will occur? And if so, why? If not, why not?
No, because because the RNA primer isn't able to do what it needs to do.
Okay.
I don't know.
Okay. So, all right. So you did good. Let me go ahead and put Ambria on the spot before I go ahead and just explain everything. So Ambria, got your mic on?
Yes.
Okay. So Ambria, just picking up where John left off. If I have glucose, right? Now, tell me first of all, bacteria, would they grow on glucose?
I'm sorry?
Will bacteria grow on glucose?
No.
They will. So a lot of the bacteria will grow on glucose, okay?
Okay.
But they can also use other things like lactose. You got me so far?
Yeah.
You with me, Ambria?
Yes.
Now, here's the question, Ambria. If glucose is present in the medium, just say we have this bacteria, right? We call it E. coli. And we put E. coli in this petri dish. And in this petri dish, we put glucose, right? If glucose is present in this petri dish, then that means my cyclic AMP levels are going to be high or low?
Low.
They're going to be low. Good, good, good. So if they're low, that means that you have this thing what we call a catabolite activator protein. We just call it a CAP, CAP, okay?
Okay.
Now, cyclic AMP, we're going to call it CAMP, CAMP. But the catabolic activator protein, we just call CAP, CAP, okay?
Okay.
So then that means that if you have glucose present, you have low cyclic AMP, it's low, and that the CAP can then bind to the activator site. You with me?
Mm-hmm.
And that can help facilitate transcription. Now, the lac repressor, that's the one that you saw that looked like the horseshoe.
Uh-huh.
It can bind to the operator site, and it can block transcription. So what are the two things we need to understand? Is that if you have CAP, right, and it binds to the activator site, that's going to help you with transcription. You follow me?
Mm-hmm.
So when I put on my cap, I'm ready to go to work. That makes sense?
Okay. Yeah, I understand.
Okay? When I got my horseshoe, when I'm putting the shoes on my horse, that means my horse ain't ready to go yet, right?
Mm-hmm.
So that means it blocks transcription. You with me so far?
Mm-hmm.
All right. Now, if you have CAP and it binds to the activator site, right? It's going to only do that if cyclic AMP is there. So if I put my hat on or my cap, once I put my cap can only stay on if I'm at camp. You follow me so far?
Mm-hmm.
Now, when glucose is there, camp is low. Didn't you tell me that?
Mm-hmm.
So if camp is low, right, then what does that mean in terms of RNA polymerase? No, you know what does polymerase mean? To grow, right?
Mm-hmm.
So poly means to extend. So that means that, so when you think about polymerase, RNA polymerase, you should be thinking about RNA growing. Okay?
Okay.
So if that's the case, then RNA polymerase cannot bind to the promoter and transcription does not occur. So once again, what do I need for CAP to happen?
For a cap to happen,
Yeah, for CAP to bind to the binding site, what do I need?
you need to be at camp.
Good. So I need cyclic AMP, right?
Mm-hmm.
But if cyclic AMP is low, can CAP bind to the binding site?
No.
So if it cannot bind to the binding site, will transcription occur?
No.
Good. So when glucose is present, cyclic AMP is what?
Low.
Low. Good. So if glucose is present, do we have transcription?
We don't.
All right. Does everybody follow that? Let me see a check, or I got John says yes. Moment says yes. Okay, I got all yeses. Okay, good. Okay, so let's keep going and let's keep following this train of thought. Okay, Moment, I'm getting back on you. You with me, Moment? Okay, so Moment, so we know what would happen if glucose is not there, right? Or if glucose is there. Okay.
Yes.
Now, let's say, Moment, glucose is present and there's no lactose. Okay. There's
Well,
no lactose. Say it again.
I'm sorry. You continue, please.
Yeah. So if you have glucose, but there's no lactose, right? Then what happens is that the lac repressor, because you don't have any lactose, right?
Yes.
So that means that the repressor, the lac repressor, will bind to the operator and block transcription. Okay?
Yes.
Now, watch this. If you have glucose and you have lactose, right? Then that means you don't have enough cyclic AMP. And if you don't have enough cyclic AMP, can CAP bind, Moment?
No.
No. So if that CAP cannot bind, we can't bind the activator site. Right?
Yes.
And if we cannot bind the activator site, now without the activator protein, RNA polymerase cannot bind to the promoter, right?
Yes.
Even though you got lactose bound to the repressor. Okay? Now watch this. Now let's say, for instance, you take no glucose and no lactose. What is your cyclic AMP going to be, high or low?
Could you repeat the question, please?
Okay. If you have no glucose, is your cyclic AMP going to be high or low?
You're freezing up on me, sir.
Oh, I'm freezing up? Okay. Let me make sure. Can you all hear me? Okay. So, Moment, what you may want to do is cut your mic off, Moment, until you're ready to talk. Okay. So, Moment, just type yes or no. Can you hear me clearly, Moment? Okay, good. So what I'll do is I'll ask you the question, Moment, and when you're ready to answer, just answer it, okay? So, Moment, the question was, if you do not have glucose, right, is cyclic AMP high or low?
It's high.
Okay, okay, very good. So he's right. It's going to be high. So if the cyclic AMP is high, then that means that the cap can bind to the activator site, right? And that means RNA polymerase can bind to the promoter. But if you don't have lactose, right, then that means that the repressor will block the operator site. And if you do that, then you will not get transcription. Does everybody understand that? Okay, good. Okay, perfect.
Lactose transcription will occur.
Say it one more time, Moment. Repeat it, Moment. Moment, repeat your question.
So, if there is no glucose but there is lactose, then transcription will occur?
Oh, okay. Okay, good. So he said if there's
Hello?
no glucose, yeah, you can cut it off. So you said if there's no glucose, but there is lactose, right? Then, of course, in that situation, no glucose means high cyclic AMP. High cyclic AMP means that cap will bind to the binding site. If it binds to the binding site, then that means that that's going to basically mean that transcription can occur. Now, in the presence of lactose, lactose will bind to the repressor. And when lactose binds to the repressor, it prevents the repressor from stopping the transcription. So transcription would occur. Everybody got that? Give me a check or agree if you got it. Okay. Okay, perfect. Okay, good deal. All right, so that is the lac operon, okay? So if you understand the lac operon, then the trip operon is a lot easier. And I'll let you kind of look at that one next time. Let's get rid of this. Okay. Let's see, embryo. What are the differences between prokaryotic and eukaryotic transcription?
Does prokaryotic use the RNA polymerase?
Mm-hmm. Well, both RNA and both prokaryotes and eukaryotes use a RNA polymerase. So that's not a difference. It is something different about that though. Not sure?
I'm not sure.
Okay. John?
Their location, eukaryotic is in the nucleus, I think.
Okay.
Prokaryotic's in the cytoplasm.
Okay. So let's look at some of the differences, okay? One of the differences is that transcription happens in the nucleus of the eukaryotes and the cytosol of the prokaryotes. The reason why is because prokaryotes do not have a membrane-bound organelle. And since a nucleus is a membrane-bound organelle, prokaryotes don't have that, okay? Transcription and translation does not occur simultaneously in eukaryotes. That means this. In a eukaryote, transcription will occur in the nucleus, and translation will occur outside the nucleus. So that means that they cannot occur at the same time. Whereas it happens outside in the cytoplasm, so these basically can happen simultaneously. Okay? The other part to that is that RNA made by the same polymerase in prokaryotes. So in prokaryotes, you have the same RNA polymerase that's doing what it needs to do in transcription to make the mRNA. So you remember mRNA is when you go from DNA to messenger RNA. So in a prokaryote, you basically have one polymerase that is responsible for that messenger RNA. Okay? Whereas in eukaryotes, which we are more complicated, we basically have 3 different polymerases that go for eukaryotes. So let me give you an example. In a eukaryote, you have a ribosomal RNA, and I'll type this so you can see it. Basically, you have, if I can type it, you have a ribosomal RNA, you have a messenger RNA, and then you have a transfer RNA. Okay? Now, basically, the ribosomal RNA is controlled by RNA polymerase 1. Okay? Messenger RNA is basically regulated by RNA polymerase 2. And then finally, transfer RNA is regulated by RNA polymerase, and I'm sure you can guess it, number 3. Now, that's a eukaryote. A eukaryote has 3, okay, Moment raised his hand. Go ahead, Moment.
Never mind. I see the typing now.
Okay. So basically, those 3 RNAs are responsible for a eukaryote. A prokaryote uses one RNA polymerase to do what it needs to do to make all 3 of these messenger RNA, or all 3 of these different types of RNAs. Everybody understand that? Okay. Messenger RNA is ready immediately in prokaryotes and needs to mature in eukaryotes. So, in a prokaryote, when you make your messenger RNA, it's already mature. It's ready to go. In a eukaryote, we're more complex. So think of a eukaryote as like starting as a baby and then growing up. In a prokaryote, they're already grown when they're born. Well, the prokaryotic messenger RNA is ready immediately. The eukaryotic messenger RNA, before it's ready, you have to allow it, you have to add what we call a poly A tail and a 5' cap. That is something we call post-transcriptional modification. We made the mRNA, it's not mature, so we need to modify it. And we modify it by adding the poly A tail and the 5' cap. Okay? I got that? Any questions? No. Sounds good. Okay, so what we're going to do is we're going to go to, let's see. Okay, everybody should be now. We're in the discussion room, and we're just going to see. If you could answer this quick poll for me, just to kind of get an idea of, we always want to improve to make sure that that we're giving you the best quality. We're missing. Okay, it's about the same. Okay, all right. So basically, we want to make sure we give you that we can communicate. The interface, I think, is a little bit better because we can add things too. The other thing that we can do is we can actually share, like if you found something on your screen or something that you have, you can actually share that with us. So if you had a document or something like that, you can actually share that document with us, which is pretty cool. Okay. Now, what we're going to do is just ask about discussion. So we talked about a lot tonight. And in the discussion notes, I'd like you to go ahead and if you have, let me see. I'm going to do something, and I want to make sure I can do it. I'm going to break you up into 2 groups, and it's going to be, and I'll put you on Bria and Donish, John and Moman, and basically you'll be in what we call a breakout room. In the breakout room, I want you to come up with 2 questions, only two questions that you think are MCAT style over any of the materials that we've covered today. Everybody understand that? And you should take about maybe 3 minutes, 3 or 4 minutes to do that, okay? Okay, so let me go ahead and set that up for you. Put you in the breakout rooms. Let's see how we get this. All right. Let's see. Start breakouts. Wait. Give me 1 second. I'll get it to second.
Ouch.
Okay. Let's see. Hey, I'm sorry. I think I messed you all up. Y'all still in breakouts?
I think we're good. Me and Mom.
Okay,
Okay.
you're good. All right, everybody's good. How was Make sure I got everybody. Um, and Bria, y'all good?
But
You are? Okay, good. Okay, perfect. Okay, so we're gonna put it on discussion board now. And what'd you say, Bria?
oh, we weren't done coming up with questions.
Oh, okay, okay. Did y'all come up with one?
Yeah.
Okay, good. All right. So what we'll do is, and Bria, if you could go ahead and put your question in the discussion notes. You can type it in.
Okay.
Okay. And so I'm assuming you're asking about the lac operon.
Yes.
Okay. Okay. So we'll open it up for, since that was your question, we'll open it up for John or Moman. Um, just raise your hand, whoever wants to answer it from your group. First one raises, you can answer it. Okay. Go ahead, John.
Transcription does not occur because glucose means that cyclic AMP is low, which means the cap will not be on, which means transcription won't occur.
Go ahead, John. Go ahead. Good job. Hold on. Let me see if I can clap. I think I can clap with that. I just gave applause. Y'all see my applause up there? That's pretty cool, huh? Can y'all do an applause, or is that just me? I can do applause. Can you do an applause? Oh, y'all can too. Okay, cool. All right. All right. Moment. How about you type your question um, into our discussion box? Moment. IO? Where's IO? Moment and John, did you all have a question?
Well, I'm sorry.
You all have a question?
Yes.
All right. Who's answering that?
Answers. You can multiple choice.
Oh, it's multiple choice. I got you.
All right. All right.
So I'm gonna alter it. So remove. Okay, I see what you did, Moment. So let me just kind of play with it a little bit. So A was that. B was that. C was that. And is he altering? I'm gonna alter. Go ahead, Donish. What's your question? Donish, you had a question? Okay, sorry about that. I'm back. Okay, did y'all answer the question? Hey, all right, is that correct? Good, perfect, 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 perfect. Okay, good job. Good job. We'll leave the breakout. Go back to the sharing. Okay, so basically that's gonna pretty much conclude it for what we're doing tonight. And so, would you all like to keep this one, or would you like to go back to the Google? Okay, so keep. I like to Google. Mom said this one. All right, Donis, you go back to Google. We're gonna stay here. All right. Okay. So I will. Your next assignment. Let me go ahead and let you know. So basically, what I want you to do is I need you to make sure that you go through your book. Even though we're highlighting some points, it's very important that you read your book. You do all the questions in your book. You got to read this book. I mean, you got to go through it. Okay, it's so important because if you want to if you want to get a 37, 38, or any of those scores, you can't just base it on class. You actually have to spend time with the book, highlighting things. If you don't understand something in the book, then tell me the page that it's on. I'll make sure that I look at it, and then we can discuss it like whenever we open up. Okay. Now, when we meet on Thursday, what we'll be covering is basically what I want you to look at is transcription translation, right? So that kind of finishes it up chapter 3. And then chapter 4 basically goes into the microbiology portion. And so I want you to look at the first part of microbiology, which talks about the viruses, all the way up into page 111. So you're going to go from like page 79 to 111, but mainly looking at the viruses and the micro, okay? But make sure you look at that for, wait, dang, tomorrow's Thursday. Oh my God. Okay. So tomorrow, what I want you to look at tonight is just the viruses. So it's on page 101 to page 101 to 111. So 10 pages. Very simple. Read the biology. Read that viruses tonight. Lysogenic and lytic cycle. Kind of understand that, and we're going to talk about that on tomorrow. Okay, everybody got me?
Yes.
Okay. All righty. Well, y'all have a great night, and I will talk to you all later. Bye bye.
Yes.